Alright people, welcome to your channel by Pankhuri Miglani. I'm Dr. Pankhuri Miglani, your biology teacher. Well, as promised, I'm standing by you in every aspect of your biology preparation for Neet 2025. Like I said, this is one-stop solution. This is the channel, this is the place, this is the teacher that you meet, that when you do a topic, it is absolutely targeting for 100% accuracy. As far as botany is concerned, I claim it, I assure you, that after watching the content, after taking classes, after taking sessions on this channel, you can sit for any kind of MCQs that have ever been asked, are going to be asked in your Neet 2025 or later. Even if you are a Neet 2026 preparation aspirant, this is the destination. Yeah, most of you who have walked through this channel know by now that I am aiming only and only at your good, okay, for your good, and I only design the sessions or plan the sessions which are beneficial for you, okay. Now, like I said in the previous class, that please go through the playlist of the channel. It's very systematic, and if you are even a little bit near your destination, you are really a NEET aspirant, okay, in your heart you know whether you are on the path of being a NEET ranker, you know that this is exactly in tune with the NEET exam, okay. Years of experience and getting students selected gives me this opportunity, I want to say, to actually know which line of NCERT, which paragraph of NCERT, which topic, subtopic, sub-subtopic could actually trap you, could lead to negative marking, and where students have superficial understanding and which leads to disaster in the NEET exam. Let's say if the exam is moderate to difficult as it was in 2022. Botany especially suffers because there are very few people who actually go into depth in a subject like botany. Okay, most of us tend to escape from it. So people, please make the use of good content, detailed content and everything given to you for your good. Alright people. Now, this is your cell cycle and cell division NCERT reading where I categorically picked up meiosis. This is sexual reproduction. More lectures will be added to this playlist. I'm going to run 11th and 12th parallelly. Genetics, like I announced in the last class of Krebs cycle, will be started post Diwali, okay, because I have to be somewhere. Although I was planning to do live lectures of principles of inheritance, but I'm going to start it. We will have very systematically, I can assure you, system is very important in your preparation. Till Diwali, I want you to finish with plant physiology. I want you to finish all these topics. We are doing respiration now. Today's lecture will be added to respiration in plants. Please share this channel, this playlist, this session with anybody and everybody who's got to do anything with any competitive exam, which includes biology, especially plant biology. And like this session so that this video remains in your live lectures and you can open it up and revise quickly at 2x or 1.5x whenever it's about Krebs cycle. And by now you know that the kind of notes that I give you, it caters to every kind of question that can be asked in your exam. Cool people, so let's start with a lot of Josh. ETC, as Janta calls it, as masses call it, electron transport chain of respirations, is quite a feared topic, okay. It is quite a scary topic for masses. Ma'am, we don't know ETC, I don't know why, but it is the most asked for topic if I do a poll or voting. Today I'm going to make it super breezy and easy for you. You will see it firsthand. Trying to complete it within 30 minutes, but for me, more important thing is to give you the power of pen. Alright, so let's start people in my own style. The terminal oxidation. Start karte hain. Please give the headings. Please write, even if you have the most awesome notes already, I want you to write with me. Terminal oxidation is what I like to call it. So now you've stepped into my world of botany, things are going to be different. Terminal oxidation is what is going to finally keep up the promise of respiration, which is giving me high amount of energy and water. But the water formed here is not the regular water. Is that clear? Carbon dioxide, which I promised in the beginning of respiration, was released in Krebs cycle. Go and watch that lecture. That is how you understand the topic. So, terminal oxidation, boys and girls, consists of, number one, the electron transport chain. It consists of electron transport chain. If I say that it is a, what does this mean actually? It means that there is a chain or a series in which electron is getting transported. In other words, it can be called as a series of oxidation, reduction, reactions. Because in chemical terms, losing electron is called as oxidation. By this, this time of your studentship, I am expecting that you know, if I lose electron, it is called oxidation. If I gain electron, it is called as reduction. So, series of oxidation reduction reactions. Where is it happening? It is happening through a series or chain of protein complexes. When I say complexes, these are some serious complexes. Usually you call them cytochrome complexes, but cytochrome is just one component of these complexes. Very important, and the easiest question of this topic is, these complexes are located in cristae. What is cristae? The inner membrane of mitochondria. You study that in the chapter of cell. There is no problem. So, the site of terminal oxidation, if they ask you, it is cristae or the inner mitochondrial membrane. These complexes consist of cytochromes, which you anyways know that, yeah. The names of the complexes, those of you who know, are cytochromes, right? Then there is iron sulfur. Very, very important. Iron sulfur is one of the major components. Then there are cofactors. Then there are other cofactors. But as such, everything is properly visible and audible. But I would love to have a feedback from you guys. Yeah, what topics you want, what more do you need in the class? Yeah, whatever it is, just open your heart out in the comment section and I'll be happy to include that. All right, people. Now, this series of, or the transport of electrons through this chain of cytochromes, which we are going to study today, ultimately leads to the formation of metabolic water. Okay? This is one of the promises of respiration, that we will form water, but this is metabolic water. Okay? The same thing that I told you, kangaroo rat. Then what happens, people? The second aspect, or the other interlinked aspect, I repeat, interlinked aspect of terminal oxidation is oxidative Photophosphorylation. What is oxidative photophosphorylation? Due to this series of oxidation reduction reactions, there is formation of ATP. Ladies and gents, in photosynthesis, ma'am taught you photophosphorylation because formation of ATP, which is simply phosphorylation, was happening in the direct presence of light. In glycolysis, I told you substrate level phosphorylation. Isn't it 7th and 10th step? The first lecture in this playlist. And this, owing to redox reactions, is called as oxidative phosphorylation. So this means that this leads to formation of ATP. Another promise of respiration is the formation of energy in the utilizable currency of cell, that is ATP. In photosynthesis, we have understood that formation of ATP within the cell will happen following the rules of chemiosmotic hypothesis. Chemiosmotic or Mitchell's hypothesis. Now, if again you are a real NEET aspirant and you are actually preparing, you know what are the requirements or components of this hypothesis, which is given in NCERT. It says that for formation of ATP, I require a semi-permeable membrane because it's osmosis, chemiosmosis, isn't it? So, semi-permeable membrane. If you remember from photosynthesis, boys and girls, answers in the comment section if you have your heart in this. What was the semi-permeable membrane and what will be the semi-permeable membrane in case of mitochondria? Come on. If you are a real learner, then there will be the most important aspect. Across the semi-permeable membrane, I need proton gradient. Without the difference or the gradient, there is no movement, there is no formation of ATP. We also call it proton motive force, or we call it electrochemical. Please write with me so that it benefits you, and you can increase the speed of the video to at least 1.5x. Now, the force behind, the sweet force behind this formation of ATP is the coupling factor F1, F0 particle, oxysomes. We have done that in photosynthesis. So, ATP synthase or F1, F0 particles, elementary particles, oxysomes, these are the various names that you study in the chapter of cell. Okay? This is what is required in chemiosmotic hypothesis, isn't it? If you remember, in photosynthesis, plastoquinone was considered as the proton pump or the hydrogen pump. Similarly, you would require a proton pump in this case also. I need the pumping of protons to create the gradient or the difference, isn't it? We will see all this right now in today's session. Okay? So now if I say that I require a membrane, most of you know that this membrane is the cristae where complexes are located. The semi-permeable membrane that is required is nothing but the inner mitochondrial membrane. So, I am going to enlarge the inner mitochondrial membrane just like we enlarged the thylakoid membrane. Now, if you look at the structure of a mitochondria here, if you know the structure of a mitochondria, if you know the structure of a, just roughly drawing it, this is a part of the chapter cell, okay. So, this is the cristae, yeah, this is the cristae, this is the outer membrane. So, this mitochondria has matrix, this is the perimitochondrial space or PMS. Any confusions? If you don't know this, please tell me in the comment section. I will take a class and I'll explain you the chapter of cell also. Yaar. Okay, if you don't know this, just be honest and tell me what you require. But don't leave any part of the syllabus undone. That's a request here. Is that clear? So this is your perimitochondrial space, mane the space between the outer membrane and the inner membrane or the cristae. This means that this is the semi-permeable membrane which has matrix on one side and perimitochondrial space on the other side. Equate it with the stroma and lumen as I have explained. Go to that playlist of photosynthesis if you haven't watched. Just like that, the thylakoid membrane is present. Okay, so now we will do this. This is your inner mitochondrial membrane. Okay, this is your inner mitochondrial membrane. Okay, visible? We'll be very patient when it comes to this topic. Like we just saw, on one side I have the matrix, yeah, and on the other side what was present? Perimitochondrial space. Let's say this is the outer membrane, yeah, this is the outer membrane. Then this here is the perimitochondrial space. Clear, people? Like I said, you can decrease the speed of the video wherever you know that you don't know the topic. So you have to suit yourself. Now what I'm going to do is the complexes that I mentioned for electron transport chain, or let's be very specific. I like the precision in the concept. The complexes that I mentioned for terminal oxidation and not for electron transport chain only, I am going to draw them, okay, for the purpose of clarity. So let's go now. So this is your complex 1. There are 5 complexes. I am going to write the names of all complexes. So let's just draw. Then you have the complex 3. Meanwhile, this is your another entity, okay, and इसको thank God. And you know that you have some mobile carriers as well, and this is your complex 2. Okay? You have to draw with me, people. Then comes complex 3. which is also a transmembrane complex. You understand what is transmembrane? After all, these are proteins, right? In the structure of cell membrane, you study that there are two kinds of proteins. There are peripheral proteins, there are integral proteins, there are transmembrane proteins. Yes? Integral proteins, isn't it? And then we'll show you this one. Cytochrome 4, complex 4. Okay? And there is a complex 5 as well. Okay? And you know, it's raining cats and dogs in Bangalore. It's raining so heavily. I love rain, so it has been amazing. Yeah? There we go, people. So, what I'm going to do is, this is your complex 1. What is complex 1? In a while. Draw this with me. This is a mobile carrier. I'm going to name it star 1 or nothing for now. This is complex number 2. Okay? This is your complex number 3. This is a peripheral protein, like I was saying, on the surface of the membrane, isn't it? This is your complex number 4. Again, a transmembrane protein. A transmembrane protein. Okay? And this is your complex number 5. Alright, people? Very good. No problem. Now let's name these complexes. So you will give the heading as complexes of terminal oxidation. You know why I stopped for a while? Because I was about to write complexes of electron transport chain. But I changed it. I took a pause to terminal oxidation, and we will see the difference together. Okay? Ready, sab log? So the first one, complex 1, people. Trust me, book will not teach you like this. It is called as NADH dehydrogenase. So, I urge you to listen to this video carefully, revisit it, replay it till electron transport chain is so nicely studied in your head that you are no more scared of it, and whatever questions you sit for, you're like, wow, thanks to ma'am. All the questions are done. So, this is NADH dehydrogenase. Okay, people, this complex has 3 components, or rather 2 components, a flavoprotein called as FMN and FES. Okay, FMN and FES. Second complex, complex 2, let's keep a distance here. Complex 2, what is the name of the complex 2? And who is going to write the name of all the complexes? FADH2 dehydrogenase. Where was FADH2 formed in Krebs cycle? When succinic acid was changing into fumaric acid. By the help of enzyme succinate dehydrogenase. Okay? So these are the 2 names of the same complex You don't have to get scared. This has one component, which is iron sulfur. Okay? Then comes complex 3. Complex 3 is cytochrome BC1 complex. Alright, and as the name suggests, its components are cytochrome B, iron sulfur. I said that. Remember when we were talking about complexes, I said, yeah, iron sulfur is a very important component of these complexes. See? Iron sulfur. Iron sulfur. You with me? And the third one is cytochrome C1. Happy? You should be. Then comes complex 4, which is cytochrome C oxidase complex. Okay? It is called as cytochrome C oxidase complex. It has 3 components. Okay? Everything is properly visible. It is cytochrome. You should also use 2 pens at the max if you are taking down the notes. Cytochrome A. Okay? Cytochrome A3 and 2 copper centers. Okay? This is also a question. Which cytochrome has 2 copper centers? It is which complex has 2 copper centers? It is complex 4. That is why I am telling you the components. But this is not it. Complex 1 and 2, boys and girls, interact with a mobile carrier. Understand that complex 1, complex 2, complex 3 and complex 4 cannot move. They cannot move within the membrane. They are studded in the membrane at a fixed location. Once again, people, complex 1, complex 2, complex 3, complex 4 cannot move. So, I need a courier boy, as I call it, or a carrier for the electrons to carry the electrons. Because like I said, it is a series of transport of electrons, and that is why everybody says it is electron transport chain. If you want to send something from one location, from one city to another, you need a courier service. So this is the mobile carrier. Is that clear? This is the mobile carrier. In fact, there is another mobile carrier between 3 and 4, which I forgot to make. Located, NCERT says, the mobile carrier is located on the outer side of the inner membrane. This is the outer side of the inner membrane. What is the outer side of the inner membrane? Let's go to the diagram of mitochondria itself. Not going to let even a single word go above your head. Outer side of inner membrane is this. Outer side of inner membrane is this. Isn't it? That means it is located towards PMS. So I am going to draw it towards PMS. Yeah, it is located towards the outer side of the inner membrane. Now what are these carriers? Between complex 1 and 2, okay, there is a carrier called as UQ or ubiquinone. This ubiquinone interacts with complex 1 and 2, but one at a time. What does this mean? Not given in the textbook, and this is the most confusing part of ETS. Ubiquinone will interact with either complex 1 or complex 2 at a time. That means if NADH2 wants to give its electrons, then ubiquinone will interact with complex 1 and carries the electrons, transfers the electron from complex 1 to complex 3 directly. If FADH2 wants to give its electrons, that is, it wants to get oxidized, then ubiquinone is going to take the electrons from FADH2 and not NADH2. It's as simple as that when you want to send a parcel from Delhi to Bangalore, you would use a courier service which will transport your parcel from Delhi to Bangalore. But if you want to transfer your package from, let's say, Guwahati to Bangalore, then that courier service will transfer, a different courier service will be allocated to transfer the electrons from Guwahati to Bangalore. Is that clear? One at a time is what I am trying to say. Between complex 3 and 4, there is another mobile carrier which I made, and focus on that line because it will be asked in true and false statement. That is your cytochrome C. Cytochrome C. Okay. So these are your mobile carriers. Mobile carrier. The kind of movement that they do will never be flip-flop movement because that will disturb the structure of the membrane. This also you have studied in the chapter of cell. If you haven't, let me know in the comment section. I'll teach you that. It will be a lateral movement. Alright, let's do the real thing now. But before that, these 4 complexes take part in ETS. Okay, these 4, complex 1 to 4. Complex 1 to 4, let's write it here. Complex 1 to 4 will take part in the electron transport chain. In the beginning of the session, we said there is another aspect. Ye dekho, there is another aspect, oxidative phosphorylation. ETS, fine, I will do with the help of complex 1, 2, 3 and 4. Yeah? The other aspect, which is your complex 5. Complex 5 will take part, with the head and the tail that I drew, will take part in ATP formation, or in fancy terms, oxidative phosphorylation. Okay? So this complex is called as the complex 5. The complex 5 or the ATP synthase or the F1, F0 particle or the coupling factor or the oxysomes or the elementary particle. Happy? When you do a concept so nicely, when you study science in totality, how can you be not happy? You have to be happy. If you're not studying properly, you might avoid it. Some people, most of the students, 50% of the students avoid that guilt or that, uneasy feeling of understanding science superficially. Your heart knows that you're not doing enough, that you're not at the right place. Happens or not? So now let's play the game. It becomes very, very easy now. This is the complex 1 here. This complex 1 is going to help the NaDH2, which is loaded with energy. Yeah, I call it electrochemical potential gradient, right? So negative charge and positive charge will move together. Alright. So this NaDH2 will lose 2 electrons and become NAD+. These 2 electrons will go to the very first component called FMN. Remember? FMN, FES. So, FMN, and from FMN, the 2 electrons will move to iron sulfur. Please don't get scared, and at this point, drop your pens. If you're watching the video, slow down the video to normal speed, drop your pens and listen to me. I'm pacing this, okay? I'm with you. Drop your pens and listen to me. The electrons will go to the courier boy. Yeah? Will go to the courier boy, which is ubiquinone. Just like plastoquinone of photosynthesis, light reaction, non-cyclic photophosphorylation, Z scheme. If I, NaDH2, am losing 2 electrons, I am definitely going to give out positive charge as well, that is 2 protons. Since I am a transmembrane protein, I am going to pump out the 2 protons into the perimitochondrial space. Okay, I am going to give my positive charge into the perimitochondrial space. Ubiquinone, which is integral, just like plastoquinone, gets greedy on receiving 2 electrons and now wants to get stabilized. So it asks the matrix to give 2 protons. Matrix being the nice guy. I don't know if you can hear the lightning and thunder outside. Oh my God, it is so favorite of mine. Man, I love it. It's like nature talking to you. It's like nature being loud and being there, right in front of you, on your face. Yeah, we mostly forget nature and take it for granted. So these 2 protons now reduce ubiquinone to ubiquinol, just like plastoquinone. Okay? Now what happens, people? Ultimately, matrix has lost 2 protons and this continues, isn't it? This continues to happen, that perimitochondrial space will be pumped with protons and this guy will lose protons. Okay? This ubiquinone will finally realize that I cannot keep the electrons or protons for that matter. Just like plastoquinone, the Pavum guy, this ubiquinone will transfer the electrons, will have to transfer the electrons. Remember cytochrome B6F in Z scheme? To complex 3. What are the components of complex 3? What are the components of complex 3? Cytochrome B, FES, cytochrome C1. So these electrons will go to cytochrome B. From cytochrome B, they will go to FES. If you will draw the diagram, you will learn, which is seemingly complicated to you right now. I can see all of you. It's like I can just see all of you. Cytochrome C1. The problem is you guys don't study ETS like this. After complex 3 also gets positive charge from ubiquinone, this complex says, hey, what will I do with the positive charge? Right? I don't want to get reduced. I know that I'm just an electron carrier. So now these protons will be pumped into your perimitochondrial space. Once again, matrix is continuously losing the positive charge. Perimitochondrial space is becoming more charged or more acidic for that matter. Okay? So, positive, positive, positive. You see in NCERT, the polarity, the charge that is getting created across the membrane. Isn't it? Or here it will be negative, 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 negative. Now, these electrons will go to complex 4, isn't it? They will go to complex 4. What is complex 4? Cytochrome A, A3. So, they will go to, what are the components? Cytochrome A, the copper centers and cytochrome A3. This complex also pumps the positive charge into, I'm so sorry, it will first go to, it will first go to the mobile carrier. That's what I realized when I had to pump the positive charge. So these electrons will go to the mobile carrier, which is cytochrome C, located on the outer side of the inner membrane. So let's send the electrons. Arey yaar. Color is important. Even if you are using 2 colors, maintain the color scheme. So this is your cytochrome C, which will carry the 2 electrons. It will move laterally, okay, and transfer the electrons to cytochrome A and cytochrome A3. 2 electrons. This complex will also pump positive charge into the perimitochondrial space. You see, 2 to 4 to 6 protons have been pumped. Listen to every word. Okay, it's getting downloaded and then getting delivered to you. These are all questions, these are all traps, these are all negative marking wale questions, these are all ranker questions, people. And share this video because ETS is going out of the window everywhere, in every head. It is a complete muddle, if I have to say. So, for 1 molecule of NADH2 is what I am teaching you. 1 molecule of NADH2, I have got 6 protons pooled up in perimitochondrial space. From cytochrome A3, the electrons will go to the matrix. And in the matrix, these electrons are received by half molecule of water, by half molecule of oxygen, by half molecule of oxygen and 2 protons. What do you think you will get? Don't you remember photolysis of water in photosynthesis, light reaction? What will you get? You will get the much awaited metabolic water. That was the promise, metabolic water. Remember what I did in the beginning of the session. I said formation of metabolic water. I said formation of ATP. So the formation of metabolic water happens due to the terminal oxidation. Ultimately, oxygen is the final acceptor. This is also a previous year question. What is the final acceptor? Final acceptor of electrons. If you remember, in light reaction, non-cyclic photophosphorylation, NAD plus is the final acceptor. NADP plus, not NAD plus. NADP plus is the final acceptor. So, NADPH2 is formed. Similarly, oxygen is the final acceptor leading to the formation of metabolic water. Now, the second aspect, that is formation of ATP, has to happen, and in that complex 5 will take part. Remember, one problem, one cause of deficiency of matrix was ubiquinone asking for positive charge from matrix. है ना? Here also this particular positive charge that oxygen requires is also taken up from matrix. So this is another reason for the proton motive force, for the proton gradient in the chemiosmotic hypothesis, which is happening at the level of inner mitochondrial membrane this time, isn't it? So compare it or equate it with the stroma and the lumen dialogue that I taught you in non-cyclic or even cyclic photophosphorylation, isn't it? So now what happens is, due to this more acidity and the more alkalinity on this side, what will happen is the protons will finally try to go towards the matrix side, isn't it? So now these two protons, I am showing you for two protons, because for the formation of one ATP, according to the conventional concept, we require two protons. So what happens is, this is called as the F0 part of the complex 5, and this is called as the F1 part or the headpiece. ये क्या है? This is the headpiece and this is the tail. Okay, the headpiece is located, these are all questions here, these are all questions, note it down. Headpiece is located towards the matrix and tail is in the inner membrane. So these protons will now pass to the tail and they will leave from the other side. That is, they will leave into the matrix. But as they leave, the ATP synthase enzyme or the coupling factor, the ATP synthase, which is a protein, gets disturbed or undergoes a conformational change as you study in biomolecules, and this head turns, okay, as the protons enter through the tail, okay, through the foot, this head turns and it leads to the coupling reaction, the famous coupling reaction, which is ADP plus inorganic phosphate, leading to the formation of ATP. So finally I get my ATP as I promised. Okay, so both the promises of terminal oxidation have been kept. This is how you study terminal oxidation, that is ETS, terminal oxidation, which is leading to the formation of metabolic water, and oxidative phosphorylation, which is leading to the formation of ATP. Done and dusted. But not yet, you know me, I am going to talk a little, one notch further, if not two. You know what is coming up. If you have been my student, this way, that way, here and there and here, then and now and where, then you know what I'm going to be talking about. And that is the final deal, boys and girls, which is the energetics of ETC. You guys get confused in this. So, as my dharma is, I'm going to take care of the confusions. So, write down energetics of ETS. Okay? So now what happens is, first we will follow the traditional rule, what is given in NCERT. Now those of you who get wired up on, ma'am, should we follow traditional or should we follow the modern concept? Well, ask this from yourself, use your common sense. Either your question will remain the same, but the choices will be given either according to the traditional concept or according to the modern concept. ठीक है? अब what is the explanation or mechanism behind the modern concept? I have already told you the mechanism, the whole machinery behind the tradition. The machinery behind the modern concept is not in your syllabus. Why it happens? Okay? You will study that in much higher classes. Mane, if you go for post-graduation in botany or in biochemistry. Okay? So now people, if the question is asked and the choices are according to modern concept, he is not going to mix it up with the traditional concept. Either the choices will follow traditional values or the modern values. We will write both. According to the traditional concept, one ATP molecule, one ATP formation, or let's write it, formation of one ATP molecule. If you could hear the lightning and thunder, let me know in the comment section. I won't know what the mic is capturing, but it is so wonderful. Formation of one ATP requires 2 protons according to the conventional concept. I just made one ATP right in front of you. Banaya na? I just made it. Ye dekho, da 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 da, ding 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 ding. So now what will happen? Then what happens if formation of one ATP requires 2 protons? 2 protons, and we just saw that one NADH2 gives out how many protons in the PMS? 6 protons. 6 protons. ये देखो. Served on the platter. This means, therefore, one NADH2 will form how many ATP molecules? बता दो भाई. 3 ATP molecules. Alright people. Now let's calculate for FADH2. If you remember, if you see, this guy complex 2 has remained empty. And it is not explained in NCERT also, isn't it? So people skip it. But in order to, if I make a statement here that, let's say I make a statement that one FADH2, which you guys mug up very sweetly, forms 2 ATP. Yeah, these are the energetics. If you don't understand or remember these energetics, you will never be able to understand the, balance sheet of respiration. I will also do a video on balance sheet of respiration because a lot of questions are asked from there. But Baba, if you will understand glycolysis, if you will understand Krebs, if you will understand ETC, then you will understand balance sheet. Now, why do you guys jump directly to balance sheet? Appa. Can't follow the steps. You will always pay a price in life if you skip the steps. The speed or the momentum or whatever you're feeling is quite momentary. Never ever skip the steps when you're learning something. Please. Especially at the basic level. So, in order to not mug up this statement that one FADH2 is giving me 2 ATP, tomorrow you guys can say, ma'am, it's outside NCERT, ma'am. Although NCERT has shown it categorically in its diagram. It has made the use of complex 2. Okay? But in theory, it just says directly, right in front of me. It's right in, it's a ritual. NCERT is always right in front of me. So you should also have that ritual. In theory, it just directly says that FADH2 forms 2 ATP molecules. In order to explain that, what I'm going to do is, I am going to rub this. Complex 1, shoo, teeny, neeny, nee. Cutty. Let's cutty NADH2 because ma'am told us that ubiquinone will interact with one complex at a time, people. Now it is the chance of complex 2. Okay? What is complex 2? Complex 2 is FADH2 dehydrogenase. ये देखो. With the component iron sulfur. Okay? So, अब complex 2 will come into action. The electrons from, let's just rub this also. Let's make it cleaner. Very good. Very good. Okay. Very good. So, if you can, you should draw a fresh diagram for that. What is the difference? This is your complex 2. So, what will it do? It will help FADH2 to get oxidized into FAD+. Have you ever wondered why NADH2 was so eager on getting oxidized and why FADH2 is eager to get oxidized? Why? Think, people. If I don't form FAD+, if I don't form NAD+, and send it back to mitochondrial matrix, then how will Krebs cycle continue? Because in the last class that you watched on Mighty Sunday, we said that Krebs cycle is a series of oxidative decarboxylations. Think. So now 2 electrons will go to FES. Okay? 2 electrons will go to FES. And once again, as NADH2 was getting oxidized to NAD+, yeah, it was also pumping the positive charge. Look at the diagram, to PMS, perimitochondrial space, because it was a transmembrane protein. It was a transmembrane protein. It was a transmembrane protein. Complex 2 is an integral protein and it cannot transport. It cannot transport the positive charge to the perimitochondrial space. Is that clear? Very clear? Ubiquinone will use those protons, will use that positive charge and get reduced to ubiquinol. Isn't it? It will take the 2 electrons. Ultimately, the same story, it will give the positive charge and the negative charge to complex 3. Complex 3 will be like, are you crazy? I am not going to keep the positive charge, Baba. It is going to pump that positive charge. Wow, man, we have never understood it this way. Aya, perimitochondrial space, and the game continues. Rest of it will remain the same. This mobile carrier, cytochrome C, will transport to complex 4. Protons will be pumped, positive charge will be pumped out, 4 protons, ultimately ATP will be formed. Actually, it's not ultimately. Complex 5 is not at the end. This is just to study. Complex 5 is a biological motor, sort of, which is active throughout. But then it's okay, at your level. It's okay, let's not confuse ourselves. But do you see the concept of 4 protons? So, ab we know what it means. Now we know. And now I am going to write it in another way because I have understood that one FADH2 leads to the pooling of 4 protons in the perimitochondrial space. Hence, or therefore, what will happen? One FADH2, one FADH2 gives rise to 2 ATP. This is the concept, traditional concept that is followed. Chamka, this much is fine. It should be. Now people, the modern concept. What is the modern concept? Very, very confusing in NCERT because NCERT has failed to change the, even your new NCERT has failed to change the text. Although in text it mentions the last line of the paragraph, it mentions that formation of one ATP, I think some page 160 or 161, formation of one ATP, right, requires 4 protons. That's all it says. Then it doesn't say anything further. It goes to sleep. Oh, 1 ATP. And actually that's the truth. This is not the truth, this one. Formation of 1 ATP does require, I mean, this is more correct as compared to this. In higher classes, you will study the mechanism, why 4 protons are pulled up. The whole transfer mechanism of ATP and everything. So, formation of ATP, 1 ATP requires 4 protons. And in diagram, it shows that 1 NADH2 pulls up 10 protons into the perimitochondrial space. Okay? So, hence, therefore, 1 NADH2 will form 2.5 molecules of ATP. 2.5 molecules of ATP from 1 NADH2. आया? चमका? अच्छा, अब क्या हुआ? Let's talk about FADH2. 1 FADH2, it was 4 protons, now it is 6 protons. In perimitochondrial space. So, therefore, 1 FADH2 will form 1.5 ATP molecules. Whew. That's how each and everything, dot to dot, beginning to end, is done for ETC. At least in this era of NEET exam, even if the paper is out of syllabus, very difficult, if you do this lecture, starting till end, write everything down, understand genuinely everything, whatever I have said, slow speed, fast speed, whatever it is, all the questions will be done. Okay? And boys and girls, let me know in the comment section anything about ETC, your doubts, apprehensions, good moments, bad moments of ETC. And subscribe to this channel, like this video, share this video with everybody. I'll see you very, very soon. Do explore the playlist and keep smiling.